Imagine this apparent paradox. Kimchi is famous for being fermented spontaneously by lactic acid bacteria naturally present on vegetables. Yet we add large quantities of salt, chili, cinnamon, garlic, ginger, black pepper and other spices at the very start. They're not just flavorings. These are natural antimicrobials. They are all proven to suppress bacterial growth. Chili contains capsaicin, and this chemical has a strong antimicrobial activity. Take a look at its chemical structure. It has this polar head group and a long hydrophobic hydrocarbon tail. So we say this molecule has an amphipathic nature. And this amphipathic nature promotes its interactions with membrane lipids. So this molecule can insert itself into biological membranes, similar to detergents that disrupt membranes. So capsaicin disrupts the outer membrane in bacterial cells, increasing its permeability. This is its antibacterial mechanism of action, but it is also known to inhibit biofilm formation. Specifically, crushed fresh garlic contains this molecule called allicin. This molecule has 2 sulfur atoms. Thanks to that, it can react with thiol groups in essential bacterial proteins, inactivating them. And as you know, bacteria have many proteins with thiol groups. So allicin in garlic attacks multiple targets within the bacterial cell. This is why bacteria cannot develop resistance to garlic. This molecule is also known to react with glutathione, a key antioxidant within the bacterial cells, and this makes bacteria sensitive to oxidative stress. So this is how garlic kills bacteria. Now what about ginger? So ginger contains this active ingredient called gingerol. So it also has a polar group and a long hydrophobic tail, similar to capsaicin in chili. So this molecule also has amphipathic nature, and its amphipathic properties also promote interactions with membrane lipids. This molecule can insert itself into biological membranes, similar to detergents that disrupt membranes. So gingerol in ginger disrupts bacterial cell membranes, increasing their permeability, and it is also known to inhibit biofilm formation. This is why ginger has antimicrobial properties. Let's take a look at why black pepper is able to kill bacteria. So the active ingredient in this case is this molecule called piperin. And it also has amphipathic structure, which promotes interactions with membrane lipids. So it also acts like a detergent. The hydrophobic part can bury itself among the fatty acid chains in the membrane lipids, while the polar part can interact with the phosphate heads at the membrane surface. So piperin disrupts bacterial cell membranes as well, increasing their permeability. But there is one additional interesting feature in this molecule, this fused ring. So this fused ring can intercalate between base pairs of bacterial DNA, and this has devastating effect on bacteria. It disrupts replication, it disrupts transcription, so this halts cell division and protein synthesis. A very potent antimicrobial compound. Cinnamon contains this interesting active ingredient with antimicrobial properties. It is called cinnamaldehyde. So it has this aldehyde group and it is super reactive. It reacts with thiol groups and amino groups in essential bacterial proteins. And as you know, bacteria have many proteins with thiol groups and amino groups, and therefore this cinnamaldehyde compound attacks multiple targets within the bacterial cell. Bacteria cannot develop resistance to it. It also has an amphipathic structure that promotes interactions with membrane lipids, so it also acts as a detergent. It has the hydrophobic part that can bury itself among the fatty acid chains of the membrane lipids, and it has a polar part, this aldehyde group, that can interact with the phosphate heads at the membrane surface, and this disrupts bacterial cell membranes, increasing their permeability. This molecule, cinnamaldehyde, is also interesting in that it has this system of conjugated double bonds, And that means that it is a flat molecule that can also intercalate between base pairs of bacterial DNA, a devastating effect on the bacterial cell that would disrupt replication and transcription. It would halt cell division and protein synthesis. So as you can see, cinnamon also has a potent antimicrobial component. And last but not the least, salt. In kimchi making, we typically use 2 to 3% concentration by weight, and this is because we know that this concentration inhibits growth of foodborne pathogens and spoilage bacteria. This makes kimchi safe. Now, this brings us to some very, very interesting questions. These antimicrobial ingredients, chili, garlic, and so on, are so appreciated for food safety. But if all these spices are able to kill bacteria, how can kimchi fermentation even start? What happens to lactic acid bacteria that are required for fermentation? And how do these antimicrobial ingredients shape the living ecosystem in kimchi? How do they shape the kimchi's taste and even the microbes in our gut when we consume kimchi? We now appreciate that the use of antimicrobial ingredients in kimchi is a double-edged sword. They make fermentation possible and safe, but they sculpt the process and outcomes in subtle and complex ways. The taste, the rate of ripening, and possible probiotic effects of kimchi are ultimately determined by how bacteria, spices, and vegetables interact with each other during fermentation. We now realize that this idea that all bacteria are equally affected by these seasonings is just wrong. These spices affect different bacteria in different ways. And these discoveries invite us to rethink not just kimchi, but how traditional fermentation technologies globally depend on a balance between selection and inhibition of microbes to achieve desired flavors, safety, and health benefits.